Hello aspirant, I hope you are doing fine. So today we are going to discuss the second part of biotechnology question in the Prelims PYQ series that we are doing from 1979 to 2024. इस सीरीज के अंदर there are the various components जो हम discuss करते हैं यहाँ पे daily basis पे. So let's see the components in this series. I am going to discuss and the team at the Edukemy going to discuss the PYQ from 1979 to 2024, means the 45 years of the PYQ we are covering here. If you see the paper of the UPSC, you will find out there is a repetition of the question or the repetition of the themes from the PYQ, so it will be the beneficial for you. Second, this series is the theme wise series. अगर आपका कोई टॉपिक वीक है इन द साइंस, मान लो बायोटेक्नोलॉजी वीक है, तो यू कैन सी द बायोटेक्नोलॉजी क्वेश्चंस फ्रॉम दिस सीरीज. अगर आपका टॉपिक इन द पॉलिटी प्रीएम्बल वीक है, तो यू कैन डू द प्रीएम्बल क्वेश्चंस फ्रॉम दिस सीरीज. दिस इज द ओनली सीरीज ऑन द यूट्यूब दैट हैविंग द थीम वाइज PYQ फ्रॉम 1979 टू 2024, जो आपको ये लिए बहुत हेल्पफुल होगा. दिस इज द ओनली सीरीज. द सेम क्वेश्चन फॉर अटेम्प्टिंग ऑन द पोर्टल, the same question of the UPSC that we are discussing here, and some extra question you will find on the Edukemy portal, and you can attempt it at free of cost. आप उसको attempt कर सकते हैं UPSC के question 1979 to 2024 in theme wise, and the link of the course is given in the description, the comment sections. And also the one more thing, the major thing, we are giving our Edukemy GS classroom notes as a complimentary to you. You can also download those notes from the comment section. Okay, so let's start the video. Let's start discussion of the second part of the biotechnology. In the first part of the video, biotechnology, we discussed the 10-12 questions. The question was mainly related to the genetic engineering, DNA, RNA. अगर हम बात करें DNA, RNA, some of the terms related to the DNA, RNA, genetic engineering. These are some specific themes from the biotechnology that we have discussed yesterday from the biotechnology, but we left some of the questions that been asked before 2010 or in between 2010 to 1979. So some of the questions we are going to discuss today, and you can also attempt all the questions of the biotechnology from the portal, from the Edukemy. The link is given in the description. Okay, so let's start with the question number 1 for today. The question number 1 is saying, what is the application of somatic cell nuclear transfer, SCNT? Even in the first video we discussed about the somatic cell nuclear transfer in the mitochondrial disease. Similarly, the somatic cell nuclear transfer can also be used in a genetic engineering for transfer of the nucleus. It can also be used for the cloning of the animals. तो आपको पता होना चाहिए कि somatic cells का कहाँ पे यूज होता है. It can be used in the cloning of animals. It can be used for the transfer of the nucleus from the one organism to the another organism. तो वहां पे somatic cell nuclear transfer होता है. Somatic cells are the cell from where we can generate the maximum cell of the human body. So somatic cell nuclear transfer है क्या? Let's read the options here. In the case of the option, the first option is production of biolarvicides, manufacturing of the biodegradable plastics, not possible. Reproductive cloning of animals, yes, this is possible, and this is the right answer also. Production of organism free of disease, no, it cannot be. And the production of the larvicide, biolarvicide, is not possible. The somatic cell nuclear transfer has a main application in the reproductive cloning of the animals. And if the question, options are similar, we need to give the most correct answer. So here the most correct answer of this question is C, the reproductive cloning of animals. So when you are reading the genetic engineering, you are reading the genetic technology, you should focus on the application of it, where we can use the technology, where we can use the biotechnology. Okay, so this was the question number 1. Now let's come to the question number 2, the genome sequencing. This is again from the genetic engineering. This is again from the genetic engineering. The here topic is saying the genome sequencing. If I am decoding the word genome, genome means gene and sequencing means the sequence of the gene. If we are doing the sequence of the gene, that is the part of genome sequencing. Now we need to be tell the genome sequencing is used in, किस में genome sequencing को use किया जा सकता है. The first statement is saying genome sequencing can be used to identify genetic markers for disease resistance. Yes, it can be used for the genetic markers. ये applications of genome sequencing है, कहाँ पे genome sequencing को use किया जा सकता है. The second, these techniques help in reducing the time required to develop new variety of crop plants? Yes. If we are doing the genome sequencing, हम disease free plants बना सकते हैं. इसके साथ साथ हम नए प्लांट का डिस्कवरी कर सकते हैं, हाई यील्डिंग वैरायटी बना सकते हैं. जीनोम सीक्वेंसिंग की वजह से देयर इज अ डिक्रीज इन द टाइम फॉर द प्रोडक्शन आल्सो. इससे मल्टीपल वैरायटी कम टाइम में बनाई जा सकती हैं. सो द सेकंड स्टेटमेंट इज आल्सो करेक्ट हियर. इट कैन बी यूज्ड टू डेसिफर in the host pathogen relationship in the crops. Yes, BT cotton, BT brinjal में यही तो किया गया. होस्ट और पैथोजन का रिलेशनशिप को जानने के लिए we are using the genome sequencing technique. So the answer of this question will be 1, 2 and 3. So the answer here will be D. If you look at this question is completely based on the applications of the genome sequencing or the application of biotechnology. But in the first video we discussed about the can be. If they are using the can be word, means this is the possibility of the doing the things कि ये possible है, or maybe it be with possible in the near future. So the answer 1 और 3 मुझे पता ही था, तो 1, 2 and 3 are the answer of the question number 2. So the answer here will be D. If I am coming to the question number 3, the question number 3 is saying in the context of the development of bioinformatics. Now the new terms bioinformatics, the term transcriptome, sometime mentioned in the news. It refers to the transcriptome term mentioned in the news. If you look at the question from the first video and the second video, we will find the two type of the question, majorly application based and specific term based. This is the question that is specifically terminology used in the biology and the biotechnology. Transcriptome is they are smaller parts that are specifically related to the messenger RNA. ये आपके पास एक factual information होना चाहिए. It is a smaller part of the messenger RNA. Let's see here. A range of enzyme used in genome editing? No. The full range of messenger RNA molecule expressed by the organism? Yes. Description of mechanism of gene expression? No. Mechanism of genetic mutation? No. It's a full range of messenger RNA expressed by an organism. When there is an expression, maybe the protein expression in an organism, that's through the messenger RNA, that completely define what is a transcriptome. तो transcriptome के बारे में define करता है. So the answer of this question will be B. So आपके लिए क्या idea रहेगा? You should focus on these terms that are used by the UPSC. And it can be repeated in the near future, maybe in the 2025. तो आपको इस तरीके की terms पे ही focus करना होता है when you are reading your biotechnology notes, biotechnology books. Like the telomeres, they can ask a chimeric DNA, they can ask the histone protein. So these are some of the terms related to it. Now let's come to the question number 4. Before going to the question number 4, at Edukemy, we are launching a large program, a very important or the most demanded program of the Edukemy, that is the Prelim Support Program for 2025 Prelims. It is not a normal test series nahi hai. It's a completely support provided to you during your preparation from December onward till the Prelims is qualified. There are various components of this program. In the last year, there are many success. There is a high success rate of this program. We'll tell you after this slide. So there are various components. The first component is the practice test and the analysis sheet. Before starting your preparation, you should know about the level of the exam. So analysis of the paper is become very important, and the practice test, you are practicing the PYQs of the UPSC, so you can help in what's the idea behind the exam creation. Examiner आपसे चाहता क्या है? Through the practice test, you can find out it out. Second, marathon session. आप लंबे टाइम तक हमारे साथ रहते हैं, 4, 5, 6 hours in a day. You are solving the question continuously. You are discussing with the teachers. Your mentors are supporting you here related to any doubts. You are making your notes. You are making your summary. Means you are sitting in the longer hours of time. So it builds your seating habits. It builds your seating habits for the longer hours. And in the UPSC time, some more most important thing, like जब 2 घंटे बैठे हैं तो आपको पता भी नहीं लगना चाहिए कि 2 घंटे आप बैठे हैं. That kind of the stamina you required in the CSE examination. Third is the book reading session. We are also doing some of the book reading sessions, like the book reading specifically the NCERT, some of the textbook like the Lakshmikant and the economy book, जहाँ पे आपको difficulty होती है, how to read the books, which portion is the important of the book, we are discussing here. Then the revision sessions after your test, book revision session, test revision session we are doing here. You are also attempting the sectional test. Each subject is having a 2 sectional tests. Sectional test में specifically question आपके current affairs से भी रहते हैं और subject specific 80 to 85 questions आपके रहते हैं. This is having the 100 questions in this test. Then the full length test adds on the level of the UPSC. Full length test एकदम mimic करते हैं हम full length test को UPSC के level से. Then the UPSC mixed test. UPSC is also conducting the other examination like the CDS. They are also conducting the examination of the APFC. They are also conducting the examination of the EPFO. So we are taking the question from these examinations that's important for the UPSC CSC examination. How the UPSC pattern is changing. They give the highlight in these examinations because these examinations conducted before the UPSC CSC Prelims examination. So वो test भी आपको कराए जाते हैं. These are the 5-6 mixed tests we are conducting. So you are used to the every kind of the question the UPSC is asking in the different examination. Then the current affairs revision, we are taking the 25 classes of the current affairs that we are conducting the current affairs from the April 2024 to the April 2025. Means a complete full year current affairs we are discussing here. तो आपको कोई मैगजीन पढ़ने की जरूरत नहीं है, आपको कहीं वेबसाइट से पढ़ने की जरूरत नहीं है. Directly attend the class, you will get the material, read only that material, and that material will not be more than the 200-250 pages. If you are reading the magazine, that goes up to the 1500 pages. जिसको याद करना भी आसान नहीं है and return on investment भी नहीं है. Most of you are having an issue with the CSET, so we are also providing the CSET support. You can solve your doubts with the teachers, you can sit with the mentors for your doubts in the CSET, and the specific strategy for the CSET, how to solve it, that we are discussing in the classroom. Then you are also getting your personalized mentors for the specific टाइम टेबल क्रिएशन. आपको अपने डे शेड्यूलिंग में इशू है, आपको पढ़ने के तरीके में इशू है. हाउ टू अटेम्प्ट द पेपर, दैट्स द मेन idea behind this PSP program. Then the final test discussion, and you are checking your preparation level at the all India level. We are conducting the Prayas all India mock test that's are conducted by the Edukemy, and more than 1000 students are appeared in this test, so you can check your level before the UPSC. Okay, so in the last USP of PSP means the 45% of the enrolled students qualified in the last prelims. जो Edukemy के साथ enrolled थे उनमें से 45% ने prelims qualify किया. More than 50% of the question directly appeared into the examination. It's not the referral question, the similar kind of the question, the same statements, the same kind of the keywords in the question that's directly appeared. You can see these videos on the YouTube in the direct hit series. Then you can also learn, your confidence will be boost. अगर आप यहाँ पे ज्वाइन करते हैं तो आपका confidence यहाँ पे होगा when you attempt the test series, and it will boost your accuracy. If your accuracy is 30%, it can increase up to the 60 to 65% when you are attempting, or it can be goes more 70 to 75% also if you are doing it religiously. So these are the USP of the PSP. Okay, I hope आपको समझ आएगा program. If you want to join the program, you can go to the Edukemy website, or you come to the ORN center. You can join the program from there. Also, a link is given in the description for the test series. You can join there. Okay, now let's come to the question number 4. The question number 4 is about the Genetic Engineering Appraisal Committee, known as the GEAC. Genetic Engineering Approval Committee is the specifically committee that's doing the approval of the genetically modified crops, genetically modified organism. ये specifically control body है इंडिया में for the genetic engineering. The question is saying Genetic Engineering Appraisal Committee is constituted under which act के अंडर इसको constitute किया गया? It comes under the Environmental Protection Act of 1986. आप कुछ लोग कंफ्यूज होंगे कि GI टैग्स के साथ है, कुछ कंफ्यूज होंगे कि वाइल्ड लाइफ एक्ट के साथ है, बट the answer is Environmental Protection Act of 1986, and it comes under the Ministry of Environment, Forest and the Climate Change. तो ये मिनिस्ट्री के अंदर आता है MOEFCC. तो ये भी आपको पता होना चाहिए which ministry heading this body GEAC. So that answer is the Ministry of Environment. Okay, so that's a very simple question. अगर आपको फैक्ट थोड़े बहुत याद हैं तो आप इसको डायरेक्टली अटेम्प्ट कर सकते हैं. Now let's come to the question number 5. So question number 5, the Genetic Engineering Approval Committee permission is required for the cultivation is under which ministry? That we discussed कि किस ministry के अंदर आता है? That's the Ministry of Environment and Forest. So the answer here will be B for the question number 5. Very simple question, direct question. This is a pattern of the UPSC, they can repeat. But nowadays, they are, instead of asking a direct question, they will give one statement out of the 3 statements in the question. This comes under the Ministry of Environment, Forest and the Climate Change. So the answer is here, Environment Ministry. The answer will be B. Now let's come to the question number 6. The question number 6 is saying, which of the following approaches comes under the category of biotechnology? There are a few statements are given. Out of these statements, which are coming under the biotechnology. Let's read the statement. The first statement is saying use of living organisms, so substance obtained from the industrial process. Yes, we're using it. Modernizing the commercial industry to produce products of use in biological research. Okay. Use of modern technology to investigate biological disorder. Yes. Use of industrial technology to increase the biosphere. No, we are not using the industrial technology to increase the biosphere. हम नहीं use करते हैं. So 4 will not be the answer. So we can say this is eliminated. This is eliminated. First, third is also correct. First and second is also correct. The answer here will be A. This is against the question based on the application कि हम biotechnology से क्या क्या कर सकते हैं. We can do the industrial process में living organism को हम produce कर सकते हैं. कमर्शियल इंडस्ट्री में बायोलॉजिकल रिसर्च में हम इसका यूज कर सकते हैं एंड बायोलॉजिकल डिसऑर्डर्स को हम इससे आइडेंटिफाई कर सकते हैं. अगेन द क्वेश्चन ऑन द एप्लीकेशन इन द साइंस एंड टेक्नोलॉजी, स्पेसिफिकली इन द यूपीएससी सीएससी प्रीलिम्स, जहाँ पे वेटेज 12 से 14 क्वेश्चन आते हैं, वो मोस्टली एप्लीकेशन पार्ट पे ही होते हैं, जो हम डे टू डे लाइफ में यूज करते हैं, इंडस्ट्री में यूज कर सकते हैं और एक बहुत बड़े ग्रुप को फायदा पहुंचाने के लिए यूज करते हैं. दैट्स काइंड ऑफ द क्वेश्चन यूपीएससी इज आस्किंग. Okay, now let's come to the question number 7. Question number 7 is saying, there's a case is given in the question number 7. If a mouse of our 8 times its normal size, if the mouse is of the one size and we are producing the 8 times size of the normal mouse, human growth hormones gene, the technique involved is क्या? इस माउस से माउस बनाने के लिए जीन चेंज होगा, तो क्या टेक्नोलॉजी मेनली रिक्वायरमेंट है? देयर आर टेक्नोलॉजी गिवन हियर. हाइब्रिडाइजेशन इज टेक्नोलॉजी फॉर द प्लांट स्पेसिफिकली, जहाँ में हम हाइब्रिड प्लांट बना सकते हैं. म्यूटेशन ब्रीडिंग इज नॉट एन आंसर. हार्मोनल फीडिंग इज नॉट एन आंसर. जेनेटिक इंजीनियरिंग इज एन आंसर. थ्रू द जेनेटिक इंजीनियरिंग टेक्नोलॉजी, थ्रू द जीन सीक्वेंसिंग, जीन चेंज करके, जीन एडिट करके हम इसको प्रोड्यूस कर सकते हैं. सो द आंसर ऑफ दिस क्वेश्चन विल बी बी. सो द क्वेश्चन नंबर 7 is B. Again the application, again the application part of technology. किस applications के through हम ये चीजें कर सकते हैं? That's again the question. Okay, now let's come to the question number 8. The sequencing of the entire genome of an organism was completed in 1996. The organism was, it's a completely factual question. फर्स्ट टाइम एक ऑर्गेनिज्म का सीक्वेंस को कंप्लीट किया गया, उसको पूरा जाना गया, उसके पूरे जीन के बारे में आइडिया हो, तो ऑर्गेनिज्म का नाम हमें बताना है इस क्वेश्चन में. इट्स एन ओल्ड क्वेश्चन, इन लाइक 1995 1996 में पूछा गया था. सो द आंसर ऑफ दिस क्वेश्चन इज द यीस्ट. आंसर ऑफ दिस क्वेश्चन इज द यीस्ट. इस क्वेश्चन ऑफ नॉट 95 96 1997 1998 के आसपास ये क्वेश्चन को पूछा गया था इन द यूपीएससी. द आंसर ऑफ दिस क्वेश्चन इज yeast. That's a factual question completely. So that's also related to the current affair perspective. उस टाइम पे कंप्लीट हो 96 में, 97 98 में क्वेश्चन पूछ लिया गया फ्रॉम द दिस ऑर्गेनिज्म जीनोम सीक्वेंसिंग. सो द आंसर ऑफ दिस क्वेश्चन इज yeast B. Now let's come to the question number 9. Again the recombinant DNA technology, that's a biotechnology, that's a genetic engineering. Allow gene to be transferred. कहाँ कहाँ और किस ऑर्गेनिज्म में जीन ट्रांसफर अलाउ करता है बायोटेक्नोलॉजी? अक्रॉस द डिफरेंट स्पीसीज ऑफ प्लांट्स. यस, इट्स अलाउड अक्रॉस द डिफरेंट स्पीसीज ऑफ प्लांट्स. तो फर्स्ट इज करेक्ट, देन द बी इज रॉन्ग हियर. फ्रॉम एनिमल टू प्लांट्स, इट्स आल्सो अलाउ फ्रॉम एनिमल टू प्लांट्स एंड इट्स आल्सो अलाउ फ्रॉम ऑर्गेनिज्म टू द हायर ऑर्गेनिज्म. सो वी कैन से द आंसर इज डी हियर. 1 2 and 3 it's allow from all the organism. In between different species also different kind of gene can be transferred here. So the question number 9 answer is D. Question number 9 answer will be D here. तो आपको देखा होगा कि again this is the application part. ये भी एक application part था about the genome sequencing, biotechnology and genetic engineering. If we are coming to the next question, the question number 10, what are the regions of the people resistance to the Bt cotton? Why the people are resisting for the Bt brinjal? Sorry, Bt brinjal. Here Bt is Bacillus thuringiensis. Bacillus thuringiensis. Similarly, in the Bt cotton, it's a Bacillus thuringiensis, and it's a Bt brinjal. Why the people are avoiding the Bt brinjals? Why the people are doing the protest against the Bt brinjal? That's a major reason. क्यों कर रहे हैं? Maybe the जो major concern मुझे लगा, that's a health concern to the people. the gene edited things, gene editing crops are not supposed to be having the positive effects, they are also having the negative effects on some human beings, जिसकी वजह से some humans are affecting it. Also it's also affecting the environment. In the case in the pollination, it's also affecting the nearby crops, that's the non-genetic crops. That's why the people are against the genetic engineering crops, or we can say the BT brinjal, or maybe some other kind of the crops. Okay, so let's read the statements. The first statement is saying BT brinjal has been created by inserting a gene from a soil fungus. It's a wrong statement. It's not from the soil fungus, it is from the bacteria Bacillus thuringiensis. Bacillus thuringiensis से हुआ है, soil fungus से नहीं हुआ. So the first statement is not correct. Let's see the option. Here the first is right given, wrong. Here is also one is included. Even in the D, option number 1 is included. So A, B and D are not the answer. So we are left with the only option number C, that is the 3 and the 4. So if I know only the one fact, directly the question is solved. आपको आगे पढ़ने की जरूरत भी नहीं है. So this is the smart work you can do in some of the question if you know the things. एक या दो स्टेटमेंट भी पता है आपको, तो आप डायरेक्टली ऑप्शन से जाके भी उसको सोल्व कर सकते हैं. एंड द पैटर्न इज अगेन इंट्रोड्यूस इन 2024. इस तरीके का पैटर्न एलिमिनेशन का यूपीएससी ने दोबारा इंट्रोड्यूस किया आफ्टर द 2023 एग्जामिनेशन. So you can mark your answer. So I'm not reading all the statements, so you can read on your own and you can attempt on the portal as well. Now let's come to the question number 11. Question number 11 is consider the following techniques and the phenomenon. Few techniques are given. Which of the above are using as the transgenic crops? ट्रांसजेनिक क्रॉप्स बनाने के लिए जेनेटिक इंजीनियरिंग का यूज होता है, जीन सीक्वेंसिंग एंड जीन एडिटिंग का यूज होता है. तो क्या क्या यूज किया जा सकता है? Consider the following techniques. The first is budding and grafting in the fruit plant. It's not the genetic engineering. It's a vegetative propagation के examples हैं, budding and grafting in the plants. So the first option is not correct. If I'm coming to the option, A is not correct, C is not correct. Now cytoplasmic male sterility, it can be done and it can also be done. Gene silencing can also be done. So the answer here is the 2 and 3. Again the question is based on the applications of the genetic engineering and the application of the biotechnology. So the question number 11, the answer will be B here. Again, it's the question on the application part. तो आपको लग रहा होगा कि application जब पूछा जा रहा है तो why should you focus on a different perspective? आपको application पे ज्यादा फोकस करना है with the terminology जो genetic engineering और biotechnology में यूज होता है, आपको उसके बारे में पता होना चाहिए with the DNA and RNA. Okay, now let's come to the question number 12. Now let's come to question number 12. Question number 12 is the other than resistance of the pest, what are the prospects for which genetically engineered plant have been created? किस पर्सपेक्टिव से, किस प्रोस्पेक्ट से किया जाता है कि मान लो एक तो रेजिस्टेंस ऑफ पेस्ट हो गया, ठीक है, वो वन पर्सपेक्टिव, दैट्स अ वन पर्सपेक्टिव कि पेस्ट रेजिस्टेंट वैरायटी हमें बनाना है. What are the other perspective to creating a genetic engineering plant? The second perspective can be the environmental friendly हो सकता है. Third perspective can be the drought resistance हो सकता है. There can be a different perspective. Let's read the statements. To enable them to withstand droughts, yes. To enable them to withstand droughts, let's come to the option. This is not having one, so B directly eliminated. To increase the nutritive value, yes, it can be used for increasing the nutritive value. To enable them to grow and do photosynthesis in the spaceship, ये absurd option है. Spaceship में कहाँ से इनको photosynthesis कराने के लिए किया जाएगा? That's not an idea. Increase the shelf life, that can be done. That's done in the tomato. So the answer of this question, the 1, 2 and 4, the C will be answer here of this question. Again the question is based on the application part कि कहाँ कहाँ पे application है of the genetic engineering and how it can be used. So this is the question of the biotechnology, question number 12, and we discussed the most of the question that's important for your upcoming examination of the UPSC. हमने discuss किए हैं यहाँ पे अब from the biotechnology part. So there is a pro mentor tip for you. There is a pro mentor tip for you that you should focus in the biotechnology whenever you are reading the chapter of biotechnology, the chapter on genetic engineering, the chapter on gene editing tools, DNA, RNA. तो आपको कहाँ पे focus करना है? You should focus on the DNA. You should focus on the type of the DNA. You should focus on the different technology that are based on the DNA. In the RNA, you should focus on the type of the RNA, like the messenger RNA, transfer RNA, ribosomal RNA, वायरस बैक्टीरिया में क्या है, डबल स्ट्रैंडेड सिंगल स्ट्रैंडेड आरएनए डीएनए है. उसके बाद जेनेटिक इंजीनियरिंग क्या है, जेनेटिक इंजीनियरिंग का प्रोसेस क्या है, और एप्लीकेशन ऑफ जेनेटिक इंजीनियरिंग क्या है. इसके साथ साथ आपको डिफरेंट काइंड ऑफ टर्म्स पता होना चाहिए. व्हाट इज द टीलोमियर्स, व्हाट इज द ट्रांसक्रिप्टोम, व्हाट इज द आरएनए इंटरफेरेंस, what is the different kind of the what is the tRNA, what is the circular DNA, what is the circular DNA. So different terminology आपको पता होना चाहिए. So these are terms that specifically used in the biotechnology process. Then the real time application, you will find out of the 10 questions, the 7 questions are based on the applications. So the application is the most important thing in this kind of the question, the biotechnology. तो आपको वो पता होना चाहिए, the real time application of the biotechnology and the application of a different technology like the CRISPR-Cas9 क्या technology है, उस पर भी question पूछा गया. And be specific to the topic, not a generalist कि आपको कुछ आइडिया है बायोटेक्नोलॉजी के बारे में लेकिन आपको स्पेसिफिसिटी नहीं पता है, आपको एप्लीकेशंस नहीं पता है, आपको यूजेस नहीं पता है, आपको ये नहीं पता कि और कहाँ कहाँ पे इसको यूज कर सकते हैं. दैट्स इज नॉट गोइंग टू हेल्प यू. ओके, आपको इन चीजों पे फोकस करना होगा व्हेन यू आर प्रिपेयरिंग फॉर द बायोटेक्नोलॉजी. इन द नेक्स्ट वीडियो आई एम कमिंग अप विद द इंफॉर्मेशन एंड कम्युनिकेशन टेक्नोलॉजी, जहां पे हम डिस्कस करेंगे डिफरेंट क्वेश्चन ऑफ द आईसीटी, बिकॉज द आईसीटी इज द मेजर पोर्शन ऑफ योर साइंस एंड टेक्नोलॉजी, जहां से 2 या 3 क्वेश्चन हर बार यूपीएससी पूछता है इन साइंस एंड टेक्नोलॉजी इन द प्रीलिम्स एग्जामिनेशन. Before ending the video, again reminder for you, you can also attempt the question of the biotechnology, all the question of the subjects, the theme-wise segregation from 1979 to 2024 on the Edukemy website. The link are given in the description. You can also download the science and technology GS class notes from the link given in the description, and I can assure you उसके अलावा आपको कुछ पढ़ने की जरूरत नहीं पड़ेगी. That's a document completely relevant for the prelims and the mains, having the all the PYQ answers as well as the complete class concepts with examples. You can it will be very useful for you, so you can download from the given description box, or you can also join the prelim support program that's Edukemy is launching. If you're joining the PSP program, I can assure you your chance for qualifying the prelims will increase many times than if you are not joining the program. Okay, thank you very much. Meet in the next video with the information and communication technology. Thank you.